individuals to essentially to develop the tools that would allow one to say, if we make such and such a treatment in individuals with Down syndrome, using the ideas derived from the animal research about what kinds of treatments can normalize function, to develop the tools that would allow us to say, if a child is treated in this way, have you actually improved their cognitive status and their behavioral function? And that sounds like it should be easy to do, but it's actually not that easy to do, because you must develop quite specific tests. In this case, based on what Roger just said, we know that there are some special problems with cerebellum. We know there are some special problems with this structure called hippocampus, which, by the way, is called that because it looks like a seahorse. And another part of the brain, prefrontal cortex. And these parts of the brain, which interestingly are all late developing structures in the brain. So that might be a critical hint about which parts of the brain are more affected. We have developed tests which specifically target the function of those three areas, which allow us to say, before the treatment, the given child is performing this way. Then you have the treatment, drugs that are based on the targets that these animal researchers have identified. That before the treatment they behave this way, after the treatment their performance is improved or not. And that we can do this in a statistically reliable way using sensitive neuropsychological tests. And so that's what we have contributed to this translational process of going from the animal research, which identifies the molecular and cellular targets, to the implications of those problems for specific brain areas, and then, given the functions of those brain areas, does this actually work in humans the way it looks like it works in mice? Not everything translates from mouse to human, so we need to make that extra step to accurately measure before and after. It could be a drug treatment, it could be some environmental stimulation, it could be anything, but you must be able to show in some carefully calibrated way that there's actually been an improvement, and that improvement is long-lasting. So that is our contribution, so to speak, to this entire process of going from the research lab to the patient, so to speak, taking the research all the way to the actual consequences of the cognitive and behavioral consequences of having trisomy 21. Okay.
That's all you do.
That's all I do.
Les questions sont ouvertes.
J'ai pas compris le nom de la drogue.
Ah, expérimenté par le professeur Reeves.
What's the name of it?
What's the name?
En français, si possible.
The name of
What particular drugs are you working with?
It's not a drug now.
Right.
Until it's approved for use in people, it's not a drug. And it's not approved for people.
Right.
But it's called SAG, which stands for Sonic Hedgehog Agonist.
But the mice is a mice and the man is a man, obviously. You know, so how far are we, or how far are you from experimenting on young babies or young boys or whatever?
Well, I'm a long way. But I think that what we can say in Down syndrome is that we currently have 2 clinical trials going on in the world to improve cognition in people with Down syndrome. One of those is carried out by Roche, primarily in the U.S. right now, and hopefully this year starting phase 2 in the U.S. and the U.K. And that is something that is based on findings in animal models, a drug that came out of electrophysiology, where you can take a slice of a mouse's brain and actually record how the neurons are communicating with each other. And what that showed is that there's an excess of inhibitory to excitatory input, and if you use a drug that lowers that inhibitory input to restore that balance, you completely restore the ability of a mouse to learn and remember. And that evidence was considered strong enough. There's much relative evidence, including Lynn's tests that show that this particular kind of function is what's affected in people with Down syndrome. But that was considered sufficient by Roche to invest a large
And
amount of money.
accepted by the FDA, of course. Yeah.
And accepted by the FDA.
Yeah.
The second trial is in Australia.
What is the drug?
Sorry?
What is this drug?
The experimental name is RG1662. Says 62. If you go to clinicaltrials.gov,
It's a GABA.
GABA.
Yeah.
GABA.
Yeah,
Inverse
inverse
agonist.
agonist,
Inverse
to be-
GABA.
Yeah.
which is an antagonist,
Right.
with a little more room on the bottom end. And there's another trial going on in Australia of PTZ, which has a similar effect, and again, that effect was shown in animal models, both in their learning and memory and in this electrophysiology that measures the connections of the neurons. It was shown to normalize this. That's work originally done by Fabian Fernandez and Craig Garner. And so those two things were considered sufficient basis, along with the human data, and because of these strong correlations with human data, to start these clinical trials. So I agree that a mouse is not a man. I tell my students every day, a mouse is not a man. And yet, on the other hand, the basic biology of the brain in people and in animals, other mammals, shares a lot of the basis of what goes on there. And what we really have to do is decide how far that goes. I think on the behavioral end, we have to be very careful. On the electrophysiology, we can't do that in people. So we have to make these kinds of correlations.
Sorry, same question. What's the name of the second drug?
The second drug is called PTZ,
Yeah,
which
exactly.
stands for pentylene tetrazole.
Pentylene tetrazole.
And it's acting on what?
That also restores this
Oh,
balance.
Yeah, same target.
The Roche drug, which Yann has looked at with Marie-Claude Potier, he looked at a Merck equivalent of this, which do the same thing. But the Roche and Merck drugs are very specific to the inhibitory neurons that feed into the hippocampus, whereas PTZ
More general.
is more general throughout the brain. So more possibility for side effects.
But to come to the question of the comparison between the mouse and the human, the approach that we've used is to focus not on language tasks, because obviously that doesn't apply, but to use tasks that really the same brain structure in the mouse determines that behavior as in the human, and to use tasks that are as close in the mouse and the human, and this is now possible. You can have the mice running in certain kinds of spatial mazes and the ability to do that completely depends upon the functions of the hippocampus, and then you can develop very comparable tasks for humans, for young humans, for middle age, all the way up, that require exactly the same cognitive skill. And there's a minimum of language involved, and you're just looking at the ability to find your way around in space. That's what this structure does. And so you can go very comparable task in the two species, requiring exactly the same underlying neurophysiology, and this has all been very well validated in both the animal models and in the humans. So from that point of view, and for the non-language kinds of problems, one can really make very direct comparisons between the animal models and the human case. In the language case, it's clearly more difficult. And the idea there is to try to build on what we know in general about the animal, about the parts of the brain in the animal models that are impaired, to how that might impact upon general aspects of language function. But it's more complicated in the language case.
Did you want to say more about the Merck drug? More
What
about this man-mouse issue?
The man and mouse issue. Of course, men are not mice, that's for sure. But as Lynn mentioned, also Roger, we have tests which are quite similar and that could be translated. So the effect or the consequence of the improvement of that could be translated to human. And we do that for a lot of metabolic studies, for example. This is quite obvious if you look for diabetes. The regulation of the glycemia is almost the same in the mouse and in human. And the mechanism is the same, and you can target drugs for that. But for the behavioral assessment, it's also the same. You can use paradigm, which are not exactly the same, but you can use paradigm, for example, to make the mice to recognize between a novel object and a familiar object, right? To discriminate between those. You can make the mice to learn how to have special cues to solve the how to escape from a Maurice water maze, which is amazing. Which you have it's like a swimming pool in which you have a small platform which is just underneath the level of the water.
But
And you learn.
But it will not one.
Yeah, and they will learn where to find the platform very easily. Except that in Down syndrome, mouse models are defective in that because they don't learn. So you have specific.
And children with Down syndrome are impaired in an exact equivalent of that task.
Lynn actually developed a Morris water maze for children, although he didn't throw them in a swimming pool. So he's never gone to jail. But he developed an equivalent test.
But you remember that the drug against Alzheimer, 10 years ago, 15 years ago, from Elan Pharmaceutical worked perfectly well on mice, and it failed totally on humans.
Yes,
that's why.
And there are many examples of that.
Exactly.
So you do have to be very careful about what you do.
Short question. First of all, GABA, it was a very long story, disappointing story. But what's the new data giving rationale to develop a new drug on this field? And the second question about hedgehog. Before you decide to move to human trials, and before that, you consider that the cancer risk and safety concerns is under control?
So who wants to do GABA?
Yann.
You do GABA, you do
I think for the GABA, so of course there are some concerns for the PTC because of the adverse effect to the treatment, maybe of chronic treatment, but the doses that are still in use at the moment are very low. And there are these two new compounds that come either from Merck or from Roche, which are really directing only a subunit of the GABA energy receptor. It's the alpha-5 subunit, which is a subunit which has been shown in the mouse to be important for learning and memory. So if you inactivate this gene in the mouse will not be able to learn all the tests that are based on learning and memory paradigms. And targeting this subunit allows the mice to recover even in the Down syndrome. There is a prognostic effect, which is also observed in normal individuals, normal mice. So I think the safety, and if we have been looking at the safety levels, so we have been trying to promote, for example, epilepsy using PTZ, high doses of PTZ, so this is really technical points. But we were not able to detect any side effects of this hormone. But of course, mice are not men, and they could have different metabolisms in humans that could maybe alter the response that are not seen in the mice. I think I tried to.
And the drug is injected, or is it an oral, or what?
It's injected
In the brain directly?
No, not in the brain, in the blood.
Well, for the Roche trial, it's actually
Oral.
oral.
It's oral.
Yeah. And the sonic hedgehog. So sonic hedgehog, I think, is a ways from clinical trials for some of the reasons you suggest. I don't think cancer is going to be a big issue. There are roles for hedgehog in cancer. If you increase the sonic hedgehog response for a very long time, you will dramatically increase medulloblastoma, a particular kind of brain cancer. If you look at, so we now know that many kinds of tumors have a small population of stem cells within them that actually generate all the tumor cells. Those stem cells for many kinds of cancer require sonic hedgehog stimulation for their division. So there's another pro-cancer role of sonic hedgehog. And indeed, drugs that turn down that sonic hedgehog response are under investigation for cancer treatment. However, the way we used sonic was to give a very short treatment. We gave them one injection the day they were born. We don't know exactly the kinetics of metabolism. It's another thing we need to learn before this could be used as a drug. But it's probably gone in 24 hours. And so we haven't assessed the brains completely yet, but we don't expect that there's going to be an increased cancer risk with that. As for other side effects, though, I mentioned just a few of the many, many aspects of development and now function that are maintained by Sonic Hedgehog. I think Sonic is going to be very difficult to use as a drug. If we're going to use this SAG that I talked about, we're going to have to limit administration to the areas where we want it to be effective. I don't think we can give it systemically. We need to know much more about doses, and one thing that we're involved with right now is trying to figure out what some of the, whether there are major side effects to even this very acute thing, because I would not be at all surprised. This is a very, very potent growth factor. It's used over and over and over again in development of almost every tissue type in your body. So it's not something to mess around with.
And coming back to the, I'm sorry. Yeah, coming back to the clinical trial, they are carried out on young babies, on you know, what sort of the, no.
If I may add another type of information, the Jerome Lejeune Foundation is acting as a sponsor of an additional clinical trial on babies suffering from Down syndrome from 6 months to 18 years old, having an experimental treatment for 1 year, knowing that all these usual cases are suffering from low level of thyroid hormone, and the influence of folate is also important. So we are currently promoting a clinical trial with enrolled patients, having 4 groups of parallel treatment, having placebo group, having folate, having thyroid hormone, and the combination of the 2 drugs. So there is an additional clinical trial ongoing under the responsibility of Fondation Lejeune. And this will be very soon put at the international exposure, because we plan to go to most probably US in order to enlarge the cohort of enrolled patients. We have already a set of patients, but we need 256 patients, so it's a lot, suffering from precisely Down syndrome, very young babies, and the main aim point is to assess the development capacity, acquisition of locomotor and responsiveness to interactions. It's a tricky outcome, and we have a lot of things doing at the same time. And this is different angles.
Yeah. But these other trials are on older, it's an older population from 18 and up. In fact, for various reasons, but mostly for issues of safety and FDA concern. But with some positive results there, it will be possible to march downwards to younger and younger ages, which will of course require development of appropriate measurement tools again that will be more appropriate for the younger cohort. That's not so straightforward, because 3-year-old infants and children have different cognitive capacity than 15-year-olds, 20-year-olds. So you have to sort of also develop the appropriate measurement tools in that, and that's what we're currently working on. So the future is to drive that down. But it's possible to do it at the higher end with the older individuals, and there actually the focus is as much on slowing down progression to Alzheimer's disease. So one can actually measure in the older population, 18, 20, 25, with appropriate biomarkers, you can actually perhaps detect which are the ones at greatest risk for development of Alzheimer's disease, and see if you can slow that down.
Yes, but the endpoints are not going to be the same, obviously. You know,
No, absolutely not. No, these are two different
yeah,
issues.
It's two different issues.
Two different issues, but both of them very important from a translational perspective to Down syndrome population. Since individuals with Down syndrome are now living much longer, the whole question of what happens when they are 30, 40, 50 is of great concern to parents.
But the test you mentioned before do not apply to the above 18 people.
No, sorry, they do.
They
have applicability from 11 to 30. They're quite good. We've shown the sort of psychometric properties in that age range. It's the lower age range that we're still working on. But the upper age range, these tests are quite applicable, in fact, and they've been validated in Down syndrome models, in Alzheimer models, and so on. So we're in a good position to test in that age range. but not so much in the lower age range yet. But that's really a big focus of the current research agenda.
Another question, maybe? Yes.
I'm wondering how long do you expect these products, these GABA antagonists, to be efficient in adults?
The clinical trials?
To take 1 year to have an
To come
effect?
to the market.
No, not at all.
How long would the treatment have to last?
Yeah, to be effective.
Okay.
To be effective.
Okay.
Empirical question, as they
Well,
say.
you have the data on mice.
So the mouse data shows that there are long-lasting effects, but they do attenuate over time. So there is a positive effect in the PTZ trials in your GABA alpha-5 antagonist, that after you stop the drug, you retain some of the properties, both behaviorally and physiologically, for some months, but they do attenuate. So exactly how the dosing would be done, nobody's really dealt with at this point. And what the kinetics will be in people, we think we're looking at a fairly fundamental mechanism behind the problem in neuronal communication in people with Down syndrome and in the mouse models. So if those parallels hold, it may be something that you don't have to take daily, but could be administered periodically.
Perhaps 3 months or 6 months later.
That would be a pure guess.
It's a guess, but right now the trials are based on the assumption that the dosing will not last more than a few months,
Okay.
right? Because the behavioral tests are before and after, are assuming a gap of 3 to 6 months. After that, it becomes a little harder to make easy comparisons.
So you're testing people who are not receiving the drug anymore.
Yes, there are. Yes.
Well, and a large number of people who haven't received it yet
At
to
all.
form that baseline.
We need some control groups.
Yeah, right.
Yeah. Some
And are the imaging techniques of any help, on the baby mice, as you say, just to understand what's happening in the brain when you inject the drug, say in 24 hours, that's unbelievable.
Are you doing imaging now?
Not that much to study, just to use the morphometric parameters, but not to study the processing of the drugs.
For mice, we can be a bit more invasive. So we actually we're able to put electrodes directly into the hippocampus of a living mouse, and we now have a baseline characterization of the differences between how these specific cells in the hippocampus, this is a different population of hippocampal cells, but they're the targets of this inhibitory, excitatory input. We can measure those in real time and how they communicate with each other. And we've spent about 2 years establishing that baseline, but we know what the differences are, and we're about to start dosing those animals with the drug to see if we can see in real time how quickly we get a change with that.
And here too, we can do work with humans. So we're doing 2 different kinds of neuroimaging in kids now.
Not with electrodes.
Not with electrodes, but electrodes on the head.
On
So
the
we
head.
can put, this
kind of helmet.
this helmet on the head with 64 contacts, and we're looking, actually with support from Lejeune Foundation, this particular project, we're looking for specific sort of biomarkers that are unique to Down syndrome, and then the potential for changing those as a function of the drug. So that's one approach, and we're also now beginning to use fMRI to look into specific brain regions, again, to look for some specific signature of what is different in Down syndrome compared to sort of control kids, and then to try to change that. So we can use these imaging techniques, and they're getting closer and closer in some way to what one can do in the animals. Because I don't know how much you know about sort of neuroimaging techniques, but the ways in which one can analyze the data from these is really greatly advancing to make it closer to the things that can be done in the animal models. these pattern analysis methods that allow one to look at complex sort of network interactions, even in the humans with neuroimaging, that gets closer to the types of things we can test directly in the animals. So we're kind of converging a little bit, even though you can't put electrodes directly into the brain. You can analyze the data in a much more sophisticated way to address some of the same issues that you can show in the mouse models. So there's a lot of convergence in that